The world is neatly divided into flora and fauna, plants and animals, a simple binary that comfortably encompasses most life. Yet, the kingdom fungi stands defiantly outside this dichotomy, inhabiting its own powerful and pervasive realm that is neither entirely plant nor entirely animal, but possesses characteristics aligning it more closely with the latter. For centuries, fueled by their stationary growth and root-like structures, fungi were mistakenly lumped in with botany. However, the fungi's true identity as heterotroph organisms that cannot produce their own food marked the first major conceptual shift in their classification. Unlike the elegant solar-powered efficiency of plants that perform photosynthesis autotrophy, fungi must derive their nutrition by consuming complex organic molecules created by other life forms. This consumption is achieved through a remarkably sophisticated external digestive process. Fungi excrete powerful hydrolytic enzymes, a chemical arsenal of proteases, lipases, and cellulases directly into their surrounding substrate, be it decaying wood, soil, or animal tissue. These enzymes break down the large, indigestible polymers into simpler, soluble compounds, which the fungus then absorbs through its cell walls. This mechanism of absorptive nutrition fundamentally separates them from both plants and animals, the latter of which typically ingest food internally before digestion. The genetic and structural evidence only reinforces this distinction and the kinship with animals. On a molecular level, phylogenetic studies reveal that fungi and animals share a more recent common ancestor than either shares with plants. A critical piece of evidence is the composition of the fungal cell wall. While plants are rigid due to cellulose, fungi utilize chitin, a tough, resilient, nitrogen-containing polysaccharide. Chitin is the same material that forms the tough exoskeletons of insects, spiders, and crustaceans. This shared biochemical trait is a profound indicator of their deep evolutionary connection to the animal line. Furthermore, fungal metabolism mirrors that of animals. They are aerobic organisms, consuming oxygen and releasing carbon dioxide, a process vital for energy production through aerobic respiration. This characteristic of breathing further removes them from the plant kingdom. The visible mushroom, the familiar cap and stems, merely the fleeting reproductive structure, known as the fruiting body. The main organism, the bulk of its living mass, is a hidden network of microscopic filaments called hyphae. This interwoven mass constitutes the mycelium, a sprawling, thread-like entity that infiltrates its food source. The hyphae are typically only one cell thick, maximizing surface area for enzyme secretion and nutrient absorption, making the mycelium an extraordinarily efficient biological machine. This structure allows for an intimate and often symbiotic relationship with the environment, acting as both a physical explorer and a chemical factory. The vast majority of a fungus's life is spent in this mycelial stage, often hidden beneath soil or within a substrate, demonstrating that the mushroom we see is an exception, not the rule. The fungal kingdom, therefore, represents a unique evolutionary path, fixed in place like a plant yet feeding like an animal and armored with the chemistry of an insect, establishing it as a singular and indispensable domain of life on Earth. If the forest floor is the Earth's skin, the mycelium is its nervous system, an unparalleled biological communication and transport network that operates constantly beneath the visible world, composed of myriad branching, thread-like hyphae. The mycelial web permeates the soil, decomposing logs and leaf litter with a microscopic finesse that defies human engineering. It is the vast, hidden infrastructure that binds ecosystems together, famously earning the nickname the Wood Wide Web. This analogy is particularly apt, as the network acts as a two-way information and resource superhighway, connecting individual organisms, often those of entirely different species, into a cohesive, functional superorganism. The most critical function of this web is its role in mycorrhizal symbiosis, a partnership between fungi and plant roots that is arguably the most vital mutualistic relationship on land. This ancient alliance, dating back to when the first primitive plants ventured out of the water, involves the fungus greatly extending the reach of the plant's root system. The hyphae are exponentially finer and more widespread than root hairs, allowing them to access pockets of water and essential immobile nutrients like phosphorus and nitrogen that would otherwise be beyond the plant's grasp. In exchange for this enhanced foraging capability, the plant transfers a significant portion, sometimes up to 30% of the sugars it produces through photosynthesis, directly to the fungus. This chemical payment fuels the subterranean fungal machinery. Beyond mere resource exchange, the mycelium facilitates complex cooperative behaviors that resemble intelligent communication. Through the mycelial bridges, scientists have observed the transfer of chemical signals, warning compounds, and defensive enzymes between trees. For example, when a specific tree is attacked by aphids or defoliating insects, it can send a signal through the network to neighboring trees, prompting them to begin producing defensive chemicals before they are physically attacked. This demonstrates a form of anticipatory defense. Furthermore, the network acts as a support system for stressed or shaded individuals. Large, established mother trees can preferentially transfer carbon and nutrients through the mycelium to younger, struggling seedlings growing in the deep shade of the canopy, effectively subsidizing their survival until they can reach sunlight. The sheer scale of this network is staggering. In a single cubic inch of healthy forest soil, there may be several miles of hyphal threads. This omnipresent, constantly growing, and highly conductive web confirms that the mycelium is not just a structure for feeding, but a decentralized, shared ecological intelligence that coordinates the life of the entire forest. Fungal reproduction is a process defined by a relentless exponential proliferation of spores, minuscule, single-celled entities that serve as the primary means of propagation and long-distance travel. The sheer volume of spore production is a testament to the fungi's evolutionary strategy. Saturate the environment to guarantee the survival of the species, even if the odds for any single spore are remote. The air we breathe is perpetually filled with a dense soup of these microscopic particles, estimated to be more numerous than pollen grains. A single fruiting body, such as the large fungi or giant puffballs, can produce and release trillions of spores throughout its lifespan, often creating a visible cloud of dust upon rupture or dispersal. The methods of spore release are marvels of biological physics. Gilled mushrooms, Basidiomycota, utilize a remarkable phenomenon called the Buller's drop mechanism. A small droplet of water forms and enlarges at the base of the spore. This droplet and another fluid film on the spore's surface suddenly merge, causing a rapid shift in the center of mass. This kinetic energy acts as a catapult, launching the spore horizontally away from the gill surface, effectively breaking it free from the still, humid air layer near the mushroom surface. Once launched, the minuscule mass of the sporophyll, less than a picogram, allows it to be picked up instantly by the slightest air current. This ability to leverage minimal atmospheric turbulence facilitates truly global dispersal. Spores are easily carried by updrafts and thermal currents high into the atmosphere, sometimes reaching the stratosphere. At these altitudes, they can travel thousands of miles on jet streams and atmospheric circulation cells, crossing continents and oceans. This extreme aerial distribution ensures that fungi are truly ubiquitous, ready to colonize any ecological niche, no matter how remote. This resilience to travel and exposure to extreme conditions is what gives credence to the fascinating, though speculative, idea of panspermia, given their hardiness against cold, desiccation, and even high levels of radiation. Fungal spores are considered plausible candidates for microbial life that could survive the harsh conditions of interplanetary travel, perhaps embedded in meteorites or space dust, potentially seeding life between worlds. Furthermore, the diversity in spore morphology, ranging from smooth and elliptical to spiny, star-shaped, or heavily textured, is linked to their dispersal method. Some are adapted for passing through the digestive tracts of animals' zoochore, emerging in a perfect patch of fertilizer. Others are designed to be sticky for attachment to insect bodies. But ultimately, it is the wind carrying the countless billions of microscopic, resilient fungal cells that connects virtually every point on Earth to the fungal kingdom, making the spore the ultimate terrestrial long-distance traveler and a perpetual reminder of the hidden life that surrounds us. The concept of a single, continuous organism spanning thousands of acres challenges our conventional, macroscopic view of life, typically reserved for blue whales or giant sequoia trees. Yet, the title of the largest living organism on Earth belongs to a fungus, specifically a colossal individual of Armillaria ostoyae, the honey mushroom. located in the Mala National Forest in eastern Oregon. This specimen is a prime example of the staggering, enduring potential of the subterranean mycelial network. The fungus achieves this monumental scale because of its primary lifestyle as a parasitic organism. Ostoe causes root rot, slowly killing trees by infiltrating their root systems with specialized, tough, root-like structures called rhizomorphs. These rhizomorphs, often described as mycelial shoestrings, allow the fungus to aggressively explore and colonize new territory beneath the forest floor, acting as fungal roots that protect the delicate hyphae within and facilitate efficient resource transport across vast distances. In a stable, old-growth forest like Mala, with an abundance of food, dead and dying trees, and few natural disturbances, the organism can grow untraordinary duration. The estimated age of this colossal fungus is a testament to its longevity. Scientists place the organism's age somewhere between 2,400 and 8,000 years old, making it not just the largest, but one of the longest living organisms on the planet. Its persistence demonstrates a profound evolutionary strategy: slow, continuous lateral growth rather than rapid, localized reproduction. While its thousands of ephemeral, edible mushroom caps appear above ground seasonally, the true enduring body remains hidden, steadily spreading and consuming, creating a massive biological anchor in the ecosystem. The discovery of this humongous fungus reshaped ecological understanding, forcing a revaluation of concepts of individuality, size, and longevity in nature. It highlights the immense difference between the transient fruiting body that signals the fungus's presence and the immense, enduring subterranean life form that performs the work of decomposition and nutrient cycling. The Armillaria ossoea is a silent giant, a testament to the power of decentralized life, confirming that the largest and oldest organisms on our planet are often the ones we least expect thriving in the hidden dark soil that sustains all surface life. The resilience of the fungal kingdom is perhaps most strikingly demonstrated by the discovery of thriving species within the highly contaminated environment of the Chernobyl nuclear power plant. In the aftermath of the 1986 disaster, with ionizing radiation levels remaining lethally high, researchers found various species of fungi not only surviving but exhibiting radiotropism, the tendency to actively grow towards the radiation source. Species such as Cryptococcus neoformans and Cladosporium sphaerospermum were found coating the walls inside the damaged reactor and growing on the highly radioactive graphite blocks. This astonishing feat of biological adaptation hinges on the presence of melanin, the dark pigment also found in human skin. In these radiotrophic fungi, melanin is hypothesized to function in a manner analogous to chlorophyll in plants, allowing them to perform a unique process sometimes dubbed radiosynthesis. Instead of capturing energy from visible sunlight, the melanin within the fungal cell walls appears to capture and transduce the energy from gamma radiation, while the energy conversion efficiency is likely low in a resource-scarce environment where most competitors are eliminated by the radiation. This ability gives the fungi a massive selective advantage. They are literally utilizing a destructive force as a source of sustenance. This biological breakthrough demonstrates a fundamental evolutionary flexibility and a capacity for life to find energy in the most extreme and unexpected forms. The practical implications of this research are immense and highly compelling for human technological challenges. Scientists are investigating how the radiotrophic fungi could be used to create biological shields against cosmic radiation for future astronauts undertaking long-duration space travel, such as a mission to Mars. The continuous bombardment of high-energy cosmic rays and solar particles poses a severe health risk. Fungal melanin, or even a layer of living or dried radiotrophic mycelium, could potentially be integrated into spacecraft walls or planetary habitats to absorb and convert the harmful radiation, offering a self-replicating and lightweight biological defense system. Furthermore, this extreme adaptation opens doors for microremediation of radioactive waste on Earth. The fungi's ability to tolerate, absorb, and grow in highly contaminated environments suggests they could be deployed to sequester radionuclides from soil and water, effectively cleaning up contaminated sites that are otherwise inaccessible or prohibitively expensive to remediate using conventional methods. The Chernobyl fungi serve as a powerful biological lesson that even humanity's most catastrophic events can be mitigated by nature's most resilient engineers, turning deadly radiation into a source of life and innovation. Fungi are essential pioneers in two profoundly hostile terrestrial environments: bare, nutrient-poor rock and landscapes devastated by wildfire. In both instances, they demonstrate a unique capacity to break down tough physical barriers and unlock essential nutrients, effectively creating the conditions for other life to follow. The process of soil formation is critically dependent on the fungi that form lichens. A lichen is a symbiotic organism composed of a fungus, the mycelium, usually an ascomycete, and a photosynthetic partner, the phytobiont, usually an alga or cyanobacterium. Growing directly on bare rock, often in climatically extreme regions, the lichen is a masterpiece of resilience. The fungus provides the protective structure, anchors the organism to the rock, and captures water, while the photosynthetic partner produces sugars for food. Crucially, the fungal hyphae secrete potent organic acids, most commonly oxalic acid. These acids act as natural chelating agents, chemically dissolving the minerals in the rock substrate. This slow, chemical weathering process physically breaks down the parent material, mixing it with the decaying fungal and algal biomass to form the very first thin layer of organic soil. Without this fungal engineering, the colonization of continents by vascular plants would have been impossible. The fungus, through this persistent acidic etching, lays the geological foundation for all subsequent terrestrial ecosystems. Conversely, the phenomenon of paraphilous fungi, or fire followers, showcases an opportunistic resilience in the face of ecological disaster. Species, like the prized morel mushroom Morchella, often appear in large flushes in the spring immediately following a major forest fire. While the exact trigger mechanisms are still debated, the mycelium lies dormant in the soil, and the high heat and destruction caused by the fire are necessary to shock the organism into fruiting. The heat treatment destroys competing vegetation, sterilizes the soil of some harmful pathogens, and most importantly, releases a pulse of key nutrients like nitrogen and carbon from the charred organic matter, creating a temporary, highly fertile environment perfectly suited for the fungus. The morel mycelium recognizes and responds to these post-fire conditions, dedicating a massive reproductive effort to release spores into the newly cleared and resource-rich landscape before vegetation can regrow and competition resumes. This ecological strategy ensures that the fungus can briefly dominate a niche created by catastrophe. Both the rock-breaking lichens and the fire-following morels exemplify the fungal kingdom's remarkable ability to thrive where other life fails, acting as biological pioneers that continually repair and reshape the planet's surface. Fungi are the planet's indispensable biological engine for decomposition, a role that places them at the core of all nutrient cycling. Without their activity, the Earth would become an inert graveyard, suffocated under an accumulating mass of dead wood, leaf litter, and animal remains, with vital nutrients permanently locked away from new life. Their work ensures the continuity of life by constantly retrieving and recycling the essential elements carbon, nitrogen, phosphorus that form the building blocks of all biological matter. The key to this unparalleled recycling capability lies in the fungus's mastery of enzyme chemistry. They are the only organisms, aside from a few specialized bacteria, that possess the enzymatic arsenal required to fully degrade lignin, the complex, tough polymer that provides structural rigidity to wood and makes it resistant to most other forms of decomposition. The inability of early organisms to break down lignin is historically believed to be the cause of the Carboniferous period's massive coal deposits, formed by plant matter that simply could not decompose before being buried. Fungi solved this ecological impasse. The most effective wood degraders are the white trot fungi. These species, which leave behind the white fibrous cellulose after consuming the darker, more rigid lignin, employ powerful oxidative enzymes such as leucolysis and peroxidases. These non-specific chemical agents literally dismantle the lignin molecule's complex chemical bonds, releasing carbon dioxide back into the atmosphere and unlocking the structure's embedded nutrients. Brown rot fungi. Conversely, primarily attack the cellulose and hemicellulose, leaving behind the brown, cubic cracked lignin structure, which is then often further processed by other microorganisms. This ceaseless digestive activity is not just about waste removal, it is the driving force of soil health and fertility. As the fungal enzymes break down organic matter, they convert it into humic substances and release inorganic nutrients back into the soil solution, making them available for uptake by plant roots, often facilitated by the same mycelial network. The entire food web is dependent on this recycling efficiency, from maintaining the carbon cycle by regulating the flow of carbon between the terrestrial biosphere and the atmosphere, to ensuring the continuous provisioning of nitrogen for protein synthesis. Fungi are the unsung maintenance workers whose tireless labor makes all other life possible. Their role is not just complementary, it is the absolute precondition for life on land. The incredible enzymatic power that allows fungi to decompose lignin, one of the hardest natural substances on Earth, is now being harnessed to tackle humanity's toughest synthetic pollutants through mycoremediation. This pioneering field applies the fungal kingdom's natural digestive capabilities to clean up environmental contaminants, including petroleum hydrocarbons, pesticides, industrial dyes, and even plastics. The chemical structures of many persistent organic pollutants, such as those found in crude oil and solvents, bear a striking resemblance to the complex aromatic rings found in lignin. Therefore, the same non-specific, powerful oxidative enzymes that white trot fungi evolved to break down wood can be repurposed to dismantle these man-made toxins. The white rot fungi, particularly the oyster mushroom Pleurotus ostratus, are the primary candidates for this work. When introduced to contaminated soil, a process called mycofiltration or mycofiltering, the fungal mycelium aggressively colonizes the substrate. It secretes high levels of enzymes like glycolysis, manganese peroxidases, and lignin peroxidases, which are capable of attacking and breaking the stable carbon-carbon bonds within the pollutant molecules. This process degrades the complex toxic molecules into simpler, non-toxic, and often biodegradable compounds, which the fungi can then absorb, or which are naturally broken down by other soil microorganisms. Successful pilot projects have demonstrated the effectiveness of this approach in treating oil spills. Within weeks, fungi-treated soil can show a massive reduction in total petroleum hydrocarbons (TPH) levels, effectively turning oily, black, foul-smelling earth into clean, nutrient-rich compost suitable for plant growth. The application to plastics is equally revolutionary. Certain species, notably Pestalozchiopsis microspora, have been discovered to be capable of digesting polyurethane, a ubiquitous component of modern plastics, even under anaerobic conditions without oxygen, making them ideal for treating landfill waste. The material they produce, the mycelium itself, a sustainable, biodegradable material that can be mycoremediation, offers a sustainable, cost-effective, and natural alternative to incineration or chemical treatments. by employing the fungi's billion-year-old digestive machinery. Humanity can begin to biologically process the chemical waste it has accumulated, turning environmental toxins back into components of the biosphere. Fungi occupy a precarious position in the human diet, oscillating between being celebrated as a gourmet delicacy and feared as a source of rapid, agonizing death. The culinary world cherishes species like the earthy, pungent truffles, subterranean ascomycetes, the delicate, nutty chanterelles, the robust, smoky shiitake, and the highly prized, honeycombed morels. These fungi offer not only unique textural elements, but also a profound depth of savory flavor, largely due to their high concentration of glutamates and guanolates, the key components of umami. Beyond flavor, they are nutritional powerhouses, providing complex carbohydrates, B vitamins, especially riboflavin and niacin, and important antioxidants and minerals, all with low caloric content. Yet, this bounty is shadowed by immense danger. The ease with which a delicious edible mushroom can be mistaken for a deadly one makes the fungal kingdom a minefield for the untrained forager. The most notorious and lethal fungus is the death cap Amanita phalloides. A single cap is often enough to kill an adult. It contains amatoxins, which are cyclopeptides that are undetectable by taste or smell and, cruelly, often present an initial illness that appears to subside, leading the victim to believe they have recovered. The amatoxins, however, are relentlessly attacking the liver and kidneys, often leading to total organ failure days later, requiring emergency transplantation. The threat extends beyond instant lethality. Other fungi contain poisons that cause severe gastrointestinal distress, neurological symptoms, or slow cumulative damage. For instance, species in the genus Cortinarius contain arylanine, which can cause irreversible kidney damage that manifests weeks after ingestion. To further complicate matters, many mushrooms are only toxic when consumed with alcohol, like Coprinopsis atramentaria, the common ink cap, or when eaten raw, like the morel, which must be thoroughly cooked to neutralize trace toxins. Due to this inherent duality, the identification of wild mushrooms requires rigorous knowledge of mycology, focusing on key features like spore, print color, gill structure, presence of a universal veil remnant, and environmental context. The relationship between humans and fungi for food is thus one of intense respect, caution, and profound reward, confirming the old mycological maxim, "There are old foragers," and bold foragers, but there are no old bold foragers. Fungi have evolved over millions of years into nature's most sophisticated chemists, producing a vast library of complex secondary metabolites compounds, not directly necessary for their survival, but vital for competition and defense. This chemical genius is the bedrock of fungal pharmacology, from the most crucial modern antibiotics to the most promising new mental health treatments. The discovery that revolutionized medicine globally was the accidental observation by Alexander Fleming in 1928. that the mold Penicillium notatum produced a substance that inhibited the growth of Staphylococcus bacteria. This substance, penicillin, became the first widely used antibiotic, saving countless lives during World War II and ushering in the modern age of pharmacology. Penicillin works by interfering with the synthesis of bacterial cell walls, demonstrating the fungus's power to selectively cripple competing microbial life. Beyond antibiotics, fungi offer a rich and diverse medicinal toolkit. Traditional and functional medicine systems, particularly traditional Chinese medicine (TCM), have utilized species like Reishi Ganoderma, lucidum, and Shiitake lentinula edodes for centuries. Reishi is celebrated for its triterpenoids and polysaccharides, compounds studied for their immune-modulating, anti-inflammatory, and potential anti-cancer properties. Lions. Hericium arenacos is gaining significant attention in Western research for its ability to stimulate the synthesis of nerve growth factor NGF, suggesting potential benefits for cognitive health and neuroprotection. Research suggests that the compound can temporarily loosen hyperconnected neural pathways in the brain, effectively allowing entrenched negative thought patterns and rumination to be broken or reset. When combined with psychotherapy, a single high-dose psilocybin experience can induce profound and lasting improvements in mood and outlook, making this ancient fungal compound a key frontier in psychiatric medicine. The fungal kingdom, therefore, continues to serve as an unparalleled biological pharmacy, continually offering solutions to human disease and suffering. The world of fungi harbors phenomena that border on the fantastical. Chief among them, the haunting beauty of bioluminescence, the capacity to emit cold, sustained light, referred to variously as foxfire, fairy fire, or will-o'-the-wisp in folklore. This glow is the result of an intricate chemical reaction, the oxidation of a specific molecule called luciferin, catalyzed by a fungal enzyme known as lucet. This reaction converts chemical energy directly into light energy with near-perfect efficiency, emitting a gentle, steady greenish or yellowish glow that can be bright enough to read by in the darkest environment. Over 70 species of fungi are known to be bioluminescent, including several species in the genera Mycena and Pennet. The light is often emitted by the mycelium spreading through decaying wood, a source of the foxfire lore, but can also be emitted by the fruiting body itself. The biological purpose of this ghostly light has been the subject of extensive scientific investigation and remains fascinatingly complex. The leading theory suggests that the luminescence is a clever evolutionary strategy for spore dispersal. By glowing in the dark forest, the mushrooms attract nocturnal insects such as beetles, flies, and millipedes. When these insects land on the glowing cap, they become dusted with spores, which they then carry to new locations, ensuring the fungus's propagation across the forest floor. The light is essentially an advertisement for transportation. Beyond science, the inexplicable glow of foxfire has deeply embedded itself in human culture and mythology across the globe. In various cultures, it has been associated with supernatural phenomena, guiding spirits, dangerous phantoms, or mystical energy fields. This connection of fungi to the spiritual and the mysterious is not unique to bioluminescence. Anthropological studies and interpretations of ancient art suggest a long, intimate relationship between humans and psychoactive fungi. Cave paintings in Europe and Central America, some dating back thousands of years, have been interpreted by scholars as depicting shamanic rituals involving the ingestion of fungi, highlighting their role in spiritual rites, divination, and the exploration of consciousness. The silent, glowing life of the fungal kingdom thus occupies a pivotal place in both the natural history of illumination and the deepest roots of human spiritual history. The mycelium, the vegetative core of the fungus, is increasingly recognized not just as an ecological powerhouse, but as a sustainable, versatile, and high-performance biomaterial capable of addressing humanity's most pressing challenges in construction, manufacturing, and even space exploration. This burgeoning field, often termed mycomaterials science, leverages the fungus's innate ability to act as a self-assembling biological glue. The process is deceptively simple. Fungal spores are introduced to a substrate of sterilized agricultural wastes such as hemp husks, corn cobs, or sawdust within a mold. The mycelium then rapidly colonizes the substrate, weaving a dense, highly branched network of hyphae that binds the loose particles into a solid, cohesive, and remarkably strong composite structure. Once the desired shape is achieved, the material is heat-treated to kill the organism and stop growth, resulting in a finished product that is fire-retardant, lightweight, insulating, and completely biodegradable. This process yields materials that can replace many of the environmentally damaging products currently used, such as polystyrene foam, particle board, leather, and even some plastics. The myco-architecture movement is pushing for the use of mycelium to grow bricks, wall insulation panels, sound-dampening tiles, and furniture, offering a net-zero carbon footprint solution to construction. As the material is grown on agricultural waste and can be composted at the end of its life cycle, the most visionary application, however, is in space colonization, missions to Mars, or the moon face, the challenge of transporting heavy, prefabricated building materials across vast distances. Mycelium provides a solution to this problem: in situ resource utilization. Researchers, including those at NASA, are investigating how mycelium could be grown in space using a small seed amount of fungal spores, and local resources like lunar or Martian regolith, mixed with a carbon source, perhaps cyanobacteria grown on-site. This process would enable astronauts to literally grow protective structures, self-repairing habitats, or even radiation shielding, domes utilizing the radiotrophic properties of certain fungi discussed earlier, making the fungal kingdom a key partner in establishing humanity's extraterrestrial presence. The mycelium is thus transitioning from a humble forest network to a fundamental building block of the future. To appreciate the profound significance of the fungal kingdom, one must grasp its immense antiquity. The history of fungi is measured in geological time, positioning them not as recent arrivals but as essential. Ancient architects of the terrestrial biosphere, the oldest definitive fungal fossils, identified by their chitinous cell walls and hyphal structure, date back nearly 1 billion years. This places them among the earliest complex eukaryotic life forms to exist, long predating the emergence of the first vascular plants and terrestrial animals. During the Silurian and Devonian periods, roughly 420 to 360 million years ago, fungi played a monumental, though mysterious, role in shaping the continental landscape. It is widely believed that the fungi of this era were responsible for the towering, trunk-like fossils known as prototaxites. These strange structures, standing up to 8 meters (26 feet) tall, dominated the landscape for nearly 40 million years. Scientific consensus now holds that prototaxites were the fruiting bodies or massive, coiled structures of gigantic fungi, making them the largest living things on land until the advent of true woody trees. This period of fungal dominance illustrates their early role in structural ecology and biomass aggregation. More critically, the fungi were the silent partners that enabled the entire colonization of land by plants. When the first rudimentary plant life began to transition from aquatic to terrestrial environments approximately 470 million years ago. They lacked complex root systems capable of extracting sufficient water and nutrients from the harsh, mineral-based soil. This ecological gap was bridged entirely by the fungi, which formed the earliest mycorrhizal associations. The fungi provided the massive surface area and enzymatic power needed to access the soil's resources, while the plants provided the sugars. Without this fungal bridge, the evolution of terrestrial plants, and by extension all land-based animal life that depends on them, would have been impossible. The fungal kingdom is not merely a component of the modern ecosystem. It is the ancient foundational prerequisite that allowed the entire green world to emerge and thrive, securing its place as an indispensable force throughout Earth's billion-year history. The peaceful image of fungi as mere recyclers and silent partners belies a fierce and ruthless existence at the microbial level, where many species act as cunning parasites and active predators. In the cutthroat competition for resources in soil and on other organisms, fungi employ sophisticated strategies ranging from chemical warfare to biological mind control, confirming their position as dynamic players in the natural ecosystem. One of the most dramatic examples of fungal predation involves the nematode-trapping fungi. Nematodes, or roundworms, are tiny, ubiquitous invertebrates that live in the soil. These fungi have evolved specialized mechanisms to capture and consume them. The most common trap is a microscopic, constricting ring formed by 3 cells. When a nematode attempts to swim through this loop, the cells inflate instantly upon contact, trapping the worm in a tight chokehold. Other species employ sticky knobs or non-constricting adhesive rings to capture their prey. Once immobilized, the fungus rapidly penetrates the worm's cuticle with hyphae, releasing digestive enzymes to consume the rich contents of the nematode from the inside out. This predatory lifestyle is a survival strategy, often employed when nitrogen or other essential nutrients are scarce in the fungi's primary decaying substrate, demonstrating an opportunistic shift in their foraging behavior. Even more bizarre and ecologically impactful is the phenomenon of entomopathogenic fungi that infect and kill insects. The genus Cordyceps and its anamorphic state. Ophiocordyceps is particularly notorious for its parasitic relationship with ants and other arthropods. The most famous example is the zombie ant fungus Ophiocordyceps unilateralis. When a spore of this fungus lands on a host ant, it penetrates the exoskeleton and begins to grow throughout the ant's body. Crucially, the fungus releases psychoactive compounds that hijack the ant's nervous system and manipulate its behavior. The infected ant is compelled to leave the colony and climb to a precise height on a plant stem, usually one that offers conditions of optimal temperature and humidity for fungal spore dispersal. At solar noon, the fungus forces the ant to bite down on a leaf vein or stem with a death grip, locking its mandibles in place. Once secured, the ant dies, and a fruiting bodia spore-bearing stalk erupts from the ant's head, positioning the spores perfectly to rain down onto new, unsuspecting hosts below. This level of precise behavioral manipulation is a chilling example of fungal adaptation and biological control. Furthermore, many plant diseases are caused by fungi, like rusts, smuts, and mildews, which exert enormous influence over agricultural output and natural plant populations. Conversely, fungi are also used by humans as biopesticides against invasive insects or plant pathogens. Harnessing this natural warfare for ecological benefit, from the microscopic noose of the nematode trap to the sinister, chemically induced command of the zombie ant. Fungal warfare is a constant, dramatic, and essential process that shapes the biodiversity and population dynamics of every ecosystem. Fungi are not just passive recyclers. They are active, strategic combatants. The influence of the fungal kingdom extends far beyond the local forest floor. It is a critical, yet often unquantified, biological factor in regulating global climate and atmospheric composition. Fungi act as a vast, decentralized geological and biological force, influencing the planetary cycles of carbon, water, and even cloud formation. Shutterstock, most profound climatic role stems from their participation in the carbon cycle, as detailed in Chapter 7. Fungi are the primary agents capable of breaking down the recalcitrant polymer lignin, which forms the vast majority of terrestrial biomass. This decomposition is the largest natural source of carbon dioxide released from the soil back into the atmosphere, a process known as soil respiration. Any change in fungal efficiency due to temperature shifts, changes in soil moisture, or altered forest health has direct and massive consequences for atmospheric text CO_2 concentrations. As global temperatures rise, the metabolism of soil fungi increases, potentially accelerating decomposition and releasing more stored carbon, creating a dangerous positive feedback loop where warming fuels more warming. Conversely, in permafrost regions, the thawing of ancient frozen biomass is awakening dormant fungi and microbes, initiating the decomposition of vast, previously locked reserves of carbon. Beyond their role in releasing carbon, mycorrhizal fungi Chapter 2 also play a role in carbon sequestration. By funneling carbon-rich sugars from the plant roots and incorporating them into their own massive long-lived mycelial structures and forming recalcitrant organic matter, fungi effectively lock away carbon in the soil for decades or centuries. They also contribute to the long-term sequestration of carbon through their role in mineral weathering Chapter 6. By producing organic acids, fungi chemically break down rocks, consuming text CO_2 dissolved in rainwater to aid in the weathering process, a fundamental geological mechanism that regulates atmospheric carbon over millions of years. Furthermore, the fungal kingdom is intimately connected to the water cycle and atmospheric chemistry. The mycelial network is critical for water retention and transfer within the soil, significantly impacting the drought resistance of forests. In the atmosphere itself, fungal spores Chapter 3 are believed to act as cloud condensation nuclei CCS due to their complex surface chemistry and sheer atmospheric abundance. These airborne spores provide perfect surfaces upon which atmospheric water vapor can condense to form droplets, initiating cloud and ultimately rainfall formation. This role suggests that fungal activity influences not just local ecology, but global weather patterns. In essence, the fungal kingdom functions as an enormous biological thermostat and hydro-regulator for the planet. Its billion-year-old evolutionary mastery of decomposition and symbiosis places it at a critical nexus where life and geology intersect, underscoring that the silent life beneath our feet is actively shaping the air we breathe and the climate we inhabit. The nutritional profile of fungi elevates them beyond a mere vegetable side dish, positioning them as a vital and sustainable source of protein, fiber, and micronutrients critical for human health and future global food security. While historically overlooked in nutritional science compared to plant and animal sources, the unique biochemistry of mushrooms offers significant advantages, particularly in terms of dietary fiber and amino acid composition. Mushrooms are an excellent source of protein, often containing more than vegetables like spinach or carrots, though generally less than meat or legumes. Critically, the protein content often contains many of the essential amino acids necessary for human diet, placing fungi high in protein quality. The fibrous structure of mushrooms also contributes to their substantial content of dietary fiber, which is crucial for digestive health. This fiber is predominantly chitin, the same material in their cell walls, and beta-glucans, in particular, are potent immune modulators, extensively researched for their ability to enhance the body's immune response and regulate cholesterol levels. Beyond macronutrients, fungi are dense with essential micronutrients, including a suite of B vitamins, niacin B3, riboflavin B2, and pantothenic acid B5, which are vital for energy metabolism. They are also unique among non-animal food sources for their ability to produce vitamin $. Text D_2$ ergociferol, similar to how human skin produces vitamin $. Text D_3$ upon sun exposure. The precursor compound ergosterol in mushrooms converts to vitamin $, text D_ when exposed to UV light, whether from natural sunlight or artificial lamps. This makes UV-treated mushrooms one of the only vegan sources of this crucial bone health vitamin. Furthermore, they are excellent providers of key minerals, such as potassium, which is vital for nerve and muscle function, and selenium, a powerful antioxidant. The burgeoning field of microfoods is addressing the escalating global demand for sustainable protein. Growing edible fungi, such as oyster or shiitake mushrooms, requires significantly less land, water, and energy compared to traditional livestock or even many crop-based proteins. Fungi can be cultivated vertically and rapidly on agricultural waste products, for example, spent coffee grounds, straw, offering a circular economic model that converts low-value byproducts into high-value food. This efficiency makes them a powerful tool for enhancing food security in dense urban areas and regions with limited arable land. As climate change strains conventional agriculture, the fungus offers a robust, nutritionally rich, and environmentally light solution to feeding a growing global population, reinforcing its status as a sustainable superfood of the future.